And now we'll move on and look deeper into the red blood cell in the molecule that makes up the red blood cell, and that's hemoglobin. And as I said, you have these four chains, alpha and beta chains. Each of these have in the center an iron. And the iron is metal, and that's what attracts the oxygen to the hemoglobin. Now, please understand that there isn't just one hemoglobin in each red blood cell. The red blood cell is just stacked full of these hemoglobin molecules, with each one with the capacity to carry 4 oxygen molecules on it. Makes them a very, very efficient vehicle for carrying oxygen. And also the other thing about it is the oxygen that's attached to that heme molecule is reversible. So when this red blood cell passes a cell that's in need of oxygen for cellular respiration, that oxygen can come off the heme molecule and down a concentration gradient diffuse into the cell, and then carbon dioxide can diffuse back out. Now, carbon dioxide can be carried on a red blood cell. It isn't where most of the carbon dioxide is carried. It's actually dissolved and carried in the plasma. Most of the carbon dioxide is. But there are binding sites for carbon dioxide on this heme molecule. They just happen to be different binding sites than for oxygen. This concept of cell production, or the formed elements, is hematopoiesis, but the specific idea that red blood cells are being made is erythropoiesis. Boy, that's a mouthful. Red blood cell production. So that hemocytoblast, it's the same stem cell that makes all the formed elements, and this is under the guidance of chemicals, growth factors and hormones, etc., that are differentiating the hemocytoblast and what it's putting out, whether it be a red blood cell or white blood cell. And this is based on need, and as I said, hormonal controls, is transformed into a pro-erythroblast. This pro-erythroblasts then develop into early erythroblasts. Remember from your 141 blasts build. So anytime you see that word blast, you're thinking that's a building cell. And then in terms of their development, what you have organelles in these pro-erythroblasts and the early erythroblasts. It's not until you get that the developing erythroblast then becomes a normal blast that the organelles are going to start to be expelled. So you do have ribosomes to make proteins, to make these hemoglobins. You have this accumulation of hemoglobin, so you're making more and more of them, and packing these oxygen carrying capacity protein type molecules into this red blood cell as it develops into a normal blast. And then the last thing that happens, it's the reticulocyte, which is the immature red blood cell. So immature because it isn't quite the shape, it isn't quite the capacity of a red blood cell, but it's reticulocytes that become mature erythrocytes. So you have this you have these ribosomes. That ribosomes are the site of protein production that are making these hemoglobins, filling this cell full, and also redesigning its form. So it's not until you get to the erythroblast that you have this biconcave anucleate. So the biconcave anucleate. The reticulocyte actually has expelled most of the nucleus. As a matter of fact, the nucleus is out, but it still isn't the biconcave shape of a mature red blood cell. That happens when it's released into the bloodstream. Regulation of this development of red blood cells. So when does your body know it should put out more red blood cells? Because that's quite a production. It takes a lot of energy to create all these proteins. So what will happen is you'll have tissue hypoxia. And that means the tissues are not getting enough oxygen. So too few red blood cells lead to tissue hypoxia. Too many, you're going to have blood that's too thick. So the viscosity is affected. And you have chemoreceptors for both of those, for lack of oxygen carrying capacity and for the fact that the blood becomes too thick or too viscous. The balance between the creating new red blood cells and the eliminating of red blood cells mostly is in hormonal control. And remember, the red blood cells only live about 100 or to 120 days. Certainly needs adequate supply of the raw minerals or the raw materials. Iron is one, amino acid and B vitamins, they need to be there. And then what you have is this negative feedback system, which you probably haven't heard enough about because almost everything in the body is negative feedback, is if you have blood levels are imbalanced, are not balanced in the toward hypoxia, this can be due to bleeding, or it can be due to red blood cells are dying off too fast, or it could be just in time to make some red blood cells. So understand that this happens to all of us just normally in the red blood cell production. But for some reason our tissues aren't getting enough oxygen, and the big tissue that likes oxygen is the brain, and the big tissue that likes oxygen is the heart. So blood needs to be created. And so what's going to happen is the kidney is going to get a message from the brain that it needs to release this hormone erythropoietin, which you learned about when you learned about the kidney and the juxtamedullary complex. That then is going to be released into the bloodstream. That is going to be passed on to the bone marrow. This stimulates red blood cell production in the bone marrow. That then is going to stimulate more red blood cells being produced and released into the blood, and then bring the balance back again. So erythropoietin is the major hormone that helps maintain red blood cell levels, along with the marrow, which is where the stem cells are, those the hematocytoblasts are, and then the balance can be restored. The effects of erythropoietin that increases it's more rapid maturing in the bone, increases circulating count with regard to reticulocytes, and this EPO is higher in men, but the thing about EPO, this is when you hear about blood doping, this is what's happening is that this particular complex is being increased to try to increase an individual's ability to have red blood cells, so oxygen capacity. So this particular slide doesn't really tell you anything more than the other slide did. It's just showing it in a slightly different way. And then the other thing is it's showing you a few of the variety of chemicals that are introduced to try to make you understand the whole process is very, very controlled. The life cycle of a red blood cell. And I did in this particular one take some images from some other places other than your textbook, only because I thought the images were a little easier to see. This is replacing an image that's in your book, figure 19.4, that the recycling of red blood cell. I think this one just looks a little simpler, maybe a little easier to follow. Where we left the red blood cell off with just the last slide is it was being produced coming out of the marrow of the bone, and this is the femur. So erythrocytes form in red bone marrow, so we're at the bottom of the image, closer to the right by the number 1. And then what happens is circulates for about 120 days. And the reason we pick that, I mean, we know that that's what it circulates, but these red blood cells make these pathways throughout the whole body numerous times throughout that 120 days, throughout the life. And they can start getting nicked. They can start being malformed. If they get nicked or malformed, then they're more likely to stick to each other. Blood already has this sticking capacity with regard to each other. It's part of the how a blood clot makes. It has this capacity to stick to each other. So they get retired, I guess you might say, or recycled is a better word, because it is important for your safety as a human to keep your blood from clotting. So circulating, and what actually kills them, so the phagocytosis takes place between the liver and the spleen. And one of the reasons why we say between the liver and the spleen, because the liver will then recycle the heme. The globin part, which is a plasma protein, that can also get recycled. But the heme, particularly, you're going to make sure you hold on to that iron, and the element in the liver that helps you do that is transferrin into transfer, and that then allows for the iron to be attached to a protein and then taken to the bone marrow where that production is taking place. So the heme from the liver is passed into the process back to the is recycled back to the bone marrow. A portion of the heme, bilirubin, is then placed into bile. That bile then, we know what bile does. Bile helps you emulsify fats, emulsify fat, not emulsify fats. So 120 days is recycled or broken apart between the liver and the spleen. Liver then has the capacity to take the pieces that are usable and package them so that they can go to the bone marrow, and then a portion of heme will be turned into bile or used in bile. And that's the cycling. And again, you have an excellent book, excellent image in your book, and maybe after seeing this, then the image in your book might make a little more sense to you. You do have the kidney in part of that. They're going to excrete some hemoglobin as well as a byproduct, urobilinogen, in the urine too. On to blood types. We do this in lab, and so you get a twofer with regard to this. Get this in lab. There are, on those red blood cells, which are those biconcave discs, there are proteins embedded into the membrane. Those proteins that are embedded in the membrane are recognition proteins, as well as integral proteins and peripheral proteins, but recognition proteins. And those recognition proteins, so far they've identified a couple of hundred of them, but not a couple of hundred that are problems. The only proteins so far are in a very simplistic way, the only proteins that are important are the proteins A and the proteins B. And we will pick this up on the next teaching time.